Beneath the surface of our everyday lives, a silent war is raging. It is a war fought on a microscopic scale, a battle for survival between bacteria and their viral predators, bacteriophages. These phages, as they are commonly known, are viruses that infect and kill bacteria. They are the most abundant biological entities on Earth, found everywhere from soil and water to the air we breathe, and even within our own bodies. Bacteriophages were discovered independently by two scientists in the early 20th century. Frederick Twort, a British bacteriologist, observed that bacterial colonies sometimes dissolved and disappeared, a phenomenon he attributed to a filtrable agent he believed might be a virus. Around the same time, Félix d'Herelle, a French-Canadian microbiologist, made a similar observation while studying dysentery bacteria. He coined the term bacteriophage, meaning bacteria eater, to describe the agents responsible for the bacterial death he witnessed. Bacteriophages are incredibly diverse in their structure and genetic makeup. They are much smaller than bacteria, typically measuring between 20 and 200 nanometers in size. Although their structures vary, most phages share a common morphology, consisting of a head which contains their genetic material and a tail, which they use to attach to and infect bacteria. The head is often icosahedral in shape, resembling a 20-sided die, and encases the phage's DNA or RNA genome. The tail fibers, extending from the base plate of the tail, act like probes, searching for the specific receptors that indicate a suitable host. Once the phage has attached to the bacterium, the tail contracts, injecting the phage's genetic material into the bacterial cell. They are estimated to kill trillions of bacteria every second, playing a crucial role in regulating bacterial populations in various ecosystems. In the oceans, for example, phages are responsible for a significant portion of bacterial mortality, influencing the cycling of nutrients and the structure of marine food webs. Bacteriophages are often described as precision bombers of the microscopic world, targeting specific bacteria without harming other cells. This specificity is a key advantage of phages over antibiotics, which often kill a broad range of bacteria, including beneficial ones that reside in our gut and on our skin. Once a phage has injected its genetic material into a bacterial cell, it hijacks the bacterium's cellular machinery to produce more phages. The newly produced phage components then assemble into new phages, which are released from the bacterial cell, often through a process called lysis. Lysis involves the production of enzymes that break down the bacterial cell wall, causing the bacterium to burst open and release the newly assembled phages. This rapid replication, coupled with their specificity and ability to evolve in response to bacterial resistance, makes phages formidable weapons in the fight against bacterial infections. Section 4. The Rise of Superbugs. The discovery of antibiotics revolutionized medicine, ushering in an era where bacterial infections that were once often fatal could be treated effectively. However, the overuse and misuse of antibiotics have led to the emergence of antibiotic-resistant bacteria, commonly known as superbugs. These resistant bacteria pose a significant threat to global health, as they are increasingly difficult and sometimes impossible to treat with existing antibiotics. The World Health Organization has identified antibiotic resistance as one of the biggest threats to global health, food security, and development today. Without effective antibiotics, many medical procedures, such as organ transplantation, cancer chemotherapy, and even routine surgery, become much riskier. Section 5. Phage therapy, a beacon of hope. As the threat of antibiotic resistance grows, scientists and clinicians are increasingly turning to phage therapy as a potential solution. Phage therapy offers several advantages over traditional antibiotics. As previously mentioned, phages are highly specific for their target bacteria, minimizing disruption to the body's natural microbial communities. Another advantage of phages is their ability to evolve in response to bacterial resistance. When bacteria develop resistance to a particular phage, the phage can mutate and evolve to overcome that resistance. Furthermore, phages can be discovered and isolated from the environment relatively easily. This ability to isolate phages against specific bacterial strains allows for personalized phage therapy, tailoring the treatment to the individual patient and their infection. Section 6. The challenges of phage therapy. Despite its potential, phage therapy faces several challenges that have hindered its widespread adoption. One of the main challenges is the regulatory landscape. Phage therapy is not currently approved by the US Food and Drug Administration or the European Medicines Agency as a standard treatment for bacterial infections. The regulatory framework for phage therapy is still evolving, and there is a need for clear guidelines and standardized protocols for the production, purification, and administration of phage preparations. Another challenge is the perception of phages as untested or experimental treatments. Section 7. The future of phage therapy. Despite these challenges, the future of phage therapy appears promising. The growing threat of antibiotic resistance, coupled with the unique advantages of phages, has renewed interest in this field. Research into phage therapy is expanding rapidly, with scientists exploring new ways to isolate, engineer, and deliver phages to combat bacterial infections. One promising area of research is the development of phage cocktails, which are mixtures of different phages that target a broader range of bacterial strains. Phage cocktails can help to overcome the specificity limitations of individual phages and reduce the risk of resistance development. Another area of interest is the use of genetically engineered phages, which can be modified to enhance their therapeutic properties or target specific bacterial genes involved in virulence or antibiotic resistance. In conclusion, bacteriophages, these tiny viruses with a big impact, hold immense potential in the fight against antibiotic-resistant bacteria.